All right, welcome everyone. We are very excited to be joined by Dr. Barriolhet and Dr. Patankar today for an ovarian cancer research update. Dr. Manish Patankar is a professor at the University of Wisconsin Obstetrics and Gynecology. He is a proud supporter and friend of WOCA. You can find him at many events, especially WOCA's Whisper Walk and annual Survivors Brunch. Dr. Lisa Barriolhet is an associate professor in the Department of Obstetrics and Gynecology at the University of Wisconsin School of Medicine and Public Health. She is a Dolores A. Buckner, MD, faculty fellow in gynecologic oncology and an associate professor with tenure at the University of Wisconsin School of Medicine and Public Health. She previously served as the interim chair for the Department of Obstetrics and Gynecology. Dr. Barriolhet completed her 4-year residency in obstetrics and gynecology at University of North Carolina, Chapel Hill. After finishing residency in 2009, she completed her 3-year clinical and research fellowship in gynecologic oncology at Brigham and Women's Hospital in Massachusetts. Dr. Barriolhet joined the University of Wisconsin Department of Obstetrics and Gynecology in 2012. She has been the recipient of the NCI Clinical Care Team Leadership Award and is currently a GOG Scholar Investigator. Her primary research focuses on prevention and early detection of ovarian cancer in a high-risk population. Dr. Barriolhet has published in numerous peer-reviewed journals and has been active in presenting research at national conferences. She has been recognized with multiple awards by her peers for exemplary teaching of residents and staff, as well as patient care. I would like to personally thank them both for joining us today. I know their schedules are absolutely packed, and we so appreciate all the time they take out for us. So please welcome Dr. Barriolhet and Dr. Patankar.
Thank you. Thank you for that really kind introduction. I really appreciate it. For those just joining, my camera, for reasons we'll never understand, is not working, so I'm not hiding from you intentionally. But I think my screen share is going to work just fine. I'm going to just change the presenter view a little bit here, but we should be in good shape momentarily. Can everyone see just one big slide?
Yeah, it looks good.
Perfect, thanks. So thank you for the privilege of being here. This is a really wonderful day. It's exciting to present research to patients, advocates, caregivers, loved ones. We talk to scientists day in and day out, but we don't get a chance to actually reach the people we hope will ultimately benefit the most from what we're working on. So Manish and I, as most of you know, are really close collaborators. We've been working together now for a decade, and I think we are a very good pairing in terms of bringing some clinical expertise and some true high-level knowledge of immunology, molecular biology, and organic chemistry together. So working with Manish has been one of the greatest joys of my time here as faculty, and of course working with patients. So getting to combine those two things and share it with you all is a big deal to me. So what we'll do today, we're going to start with some novel therapies and prevention strategies. That will be my portion of the presentation. And then Dr. Patankar will take over and share some slides about developing diagnostic tests. So of course, both of these things are really critical to our patients. I do want to point out, and there'll be some slides showing our team, but this is really a team effort, right? So you're going to hear me use the first person, but this is a we situation. And that we does not just include Manish and myself, but our graduate students, our postdoctoral fellows, our lab manager, our graduate, GYN oncology fellows who are training to become surgeons and researchers. So it's really the ultimate team effort. And we couldn't do this without the support of WOCA, without the support of our department and our cancer center. There's so many moving parts, and that alignment is tremendously helpful. So some of these slides I actually showed last year, but I just want to give a little context to the work we're doing. Cancer prevention is my personal area of love, and that's specific. It's not just specific to ovarian cancer. In fact, I've listed here 3 ongoing efforts at the Cancer Center that are focused on prevention rather than treatment. And one is an HPV vaccine alternative used in the context of preventing cervical dysplasia and cancer, an anti-estrogen therapy protocol protocol for preventing uterine cancer, and then what we're doing in our laboratory, which is altering cellular energy production to prevent carcinogenesis. So that brings us to a pretty complicated idea, which is how do cells turn into cancer? And this molecule I'm showing here, atovaquone, is something I think actually could be critical in preventing that process. So we won't get too into biochemistry today, but I just want to point out that normal tissue, which is shown on the left, makes energy through two pretty clear strategic pathways. And one is using part of the cell called the mitochondria. And any of you who took biology or biochemistry in college might remember learning about this. And that makes a whole bunch of ATP, and that you need oxygen. The mitochondria energy production requires oxygen. So that's what happens when oxygen's around. And when there's no oxygen, so like a skeletal muscle cell, for example, when you're exercising, loses your oxygen's being used elsewhere, might make energy through an alternate pathway, which actually produces lactic acid as a byproduct. What we've learned, and this is shown on the right about cancer cells and other proliferative tissue, is it doesn't care. They don't care if oxygen's there or not. They can switch back and forth between aerobic and anaerobic glycolysis really effectively. So we know that one of the things unique to cancer cells is this ability to be more nimble and use oxygen or not use oxygen as it sees fit. So it's really unpredictable. But when you look at some of the first changes seen in cancer cells, this is actually one of those hallmarks. And this has been described for more than 100 years. So I think focusing on these early metabolic changes gives us an opportunity to really force cells that are thinking about becoming cancerous into behaving more normally. So this brings us back to atovaquone, which I mentioned last year, and we're still studying it because I still think it holds a lot of promise. It's actually wonderful to consider as a prevention agent. And the reason for that is because it's already FDA approved. It's available in a pill, and it's part of Malarone, which is a malaria treatment and prophylaxis. So we have a ton of data about the safety, the dosing, who can take it. You know, you can even take it in pregnancy. So this is a really nice molecule to study further because so many of the pieces critical in a prevention medication, like oral availability, have already been met. So we're hypothesizing that atovaquone can halt this process of early cellular metabolic changes that can cause ovarian cancer. And what I talked about last year is I hope this could help patients, starting with those at highest risk for developing ovarian cancer. So patients with BRCA, another familial risk, would be a great place to start. This doesn't mean this couldn't be used for cancer prevention in a larger population, but in terms of studying the drug and its possible efficacy, starting with those at highest risk makes sense. And these are things we all have underway. So that's the prevention piece. I actually want to focus more on the therapeutic piece and how I think atovaquone might fit into cancer therapy. And that has to do with combination therapy. We've actually studied atovaquone as a cancer treatment, and it's just not potent enough. It definitely does some work in the Petri dish, but it's much less potent than other drugs in its class. So while I think it has some anti-tumor activity with ovarian cancer and other cancers, actually, it's not going to do the job alone. And it turns out immunotherapy, which I suspect many on this call have heard of in the context of other types of cancer, isn't doing the job with ovarian cancer either. So potentially combining the two could be a really interesting strategy, and I'll explain why. What we're showing here is T cells interacting, and that those are kind of the blue colored guys with a tumor cell. And what you need for a T cell to recognize a cancer cell is a somewhat complicated, but kind of a lock and key type of situation where the cancer cells presenting a protein or antigen that the T cell receptor says, hey, I see that. And this PD-1 and PD-L1 proteins, which we'll talk a little bit more about, they're called checkpoints, programmed cell death protein to be specific. These are connecting as well. And this is how the immune cells can recognize cancer. Turns out with ovarian cancer, this isn't happening. So in the last decade, these checkpoint inhibitor immunotherapies, which are what's shown here on the right, allow T cell killing of the tumor cell. So what this does is basically uncloak. There's some costumes that cancer cells can wear that make it harder for T cells to see it. And when those costumes are removed by a PD-L1 inhibitor, another type of immunotherapy, the T cells say, oh, wait, I see you now. You're a cancer cell. This is a problem, and I'm going to get this done and help use natural functions like your immune system to kill the cancer cell. So immunotherapy has been a huge success in cancer treatments. For melanomas in particular, certain types of lung cancers, even in uterine cancers and cervical cancers, we've seen great success. But for some reason, ovarian cancer is not interested, for the most part, in being sensitive to immunotherapy. So how do we do this? A cold tumor is a tumor that is not going to be recognized by the immune system. Can we turn it hot? Can we make it more susceptible to immunotherapy? So there is a building body of evidence that there are mechanisms to make cold tumors hot. And one of these approaches that we know of is radiation therapy. So we know that radiation therapy, when combined with checkpoint inhibitors or immunotherapy, can be more effective as combination. So it seems that some of the changes radiation causes can actually make immunotherapy work better. Great. But radiation therapy, as many of you know, is not the mainstay of ovarian cancer treatment. Ovarian cancer typically is throughout the abdominal cavity on multiple sites, and you cannot radiate the entire abdominal cavity without experiencing some really intense and unpleasant side effects. But we do know that in a petri dish where you can use radiation at liberty, that ovarian cancer cells are sensitive. So it's a way that we can recreate some of the cellular damage we see after radiation using other things like atovaquone. And we think the answer might be yes. So what atovaquone actually does is block something called oxidative phosphorylation, and that causes intracellular oxidative stress. So that can lead to DNA and protein damage and ultimately cell death. So could we prime the tumors, the ovarian cancer cells, with atovaquone and make them more effective to immunotherapy? So I'm showing some data of part of a poster presentation that was presented at the American Association for Cancer Research last week by our MSTP, which is our MD PhD student, Myra Bentoncourt-Ponce, who's awesome. This is some of her work that I'm showing. And this is really her thesis, is what can we do to fire up the immune system and get it to work more effectively in ovarian cancer? And she thinks that atovaquone can increase reactive oxygen species and oxidative stress and cause a few different things that are really important to getting the immune system to work effectively. So I've already mentioned DNA protein damage. I'm going to talk a little bit about something called the unfolded protein response and also the release of DAMPs. DAMPs are damage-associated molecular patterns, and these are released from damaged or dying cells. They activate the innate immune system. So we think a combination of these things would increase ovarian cancer immunogenicity and hopefully lead to improved T cell recognition and the elimination of tumor cells. So that's basically what I just said. So this combination idea will boost immune response. The next slide, which I won't go into great detail, just shows that these DAMPs are part of a really complex interaction between innate immune cells, of which there are many. So these are things just circulating around in our body, ready to be activated the minute the cold or flu virus infiltrates, the minute a foreign body decides to lodge under our skin like a splinter. These are all things that are kind of at the ready. Non-immune cells are the types of cells, and cancer cells are included here, that may release other inflammatory mediators or other things that interact with the immune system. And then adaptive immune cells are T cells and B cells. They're going to they're really going to have a more specific response to whatever it is they're being faced with. So I just put DAMPs in the middle here to show this complex interaction when we think about immune cell activation. So this is more of Myra's data. And what she did was say, hey, what's happening with DAMPs? What's happening with these damage-associated molecular patterns in ovarian cancer cells after they're treated with atovaquone? Does that make them more active, less active? What are we seeing? So HMGB1 and TFAM are both examples of DAMPs. And you can see here, this is ovarian cancer cell line. So OVCAR3 is a human ovarian cancer cell line that we keep in culture. There are many different ovarian cancer cell lines that we're studying. This is just one example, and we often will duplicate or replicate the same experience in multiple cell lines to best understand who they may be most likely to help. And you can see that the y-axis here to the right is going to show increasing exposure to atovaquone over time. So on the left, we have a pretty short exposure, which is 8 hours. Then we have 24 and 48 hours moving towards the right. And the IC50 and IC75 are different concentrations. So the higher concentration is the IC75. So you can see that with increased duration and increased concentration, we're seeing more damp release from ovarian cancer cells. So that does indicate that atovaquone may be activating a small part of the immune system. So other things she studied was, hey, what is this doing to reactive oxygen species, which is that's the same piece of the puzzle that we know radiation activates. So radiation will also increase reactive oxygen species, and we think make cells more susceptible to immunotherapy. Can we see some of that same increase with atovaquone? Again, OVCAR3 cells, atovaquone treatment, and we did see the shift to the right, which is a shift showing increased ROS levels. So there's an indicator, and it's the alpha H2AX indicator that is becoming increased after this DNA damage occurs. So this basically is the H2AX is a marker of double-strand DNA breaks. So we see more of that after atovaquone exposure. Now, I briefly mentioned the endoplasmic reticulum, which is actually very beautiful when seen schematically. It's kind of this complicated labyrinth of cisterna that also exists inside the cells. The endoplasmic reticulum is the principal organelle responsible for a ton of different cell functions, including protein folding, the maintenance of cellular hemostasis, protein maturation. And ER stress is activated by a variety of factors and triggers something called the unfolded protein response, the UPR. And the UPR restores homeostasis or activates cell death, depending on the situation. Multiple studies have clarified the link between ER stress and cancer, and particularly this UPR, this unfolded protein response, is something, again, in the carcinogenesis pathway. So Myra also looked at this, ER stress effectors. So these, the ATF2 and DDIT3 are both downstream effectors of ER stress. So just pretty basic ways that we can measure that response. And atovaquone treatment, same thing. More higher concentrations and more time led to more expression of these ER stress pathway effectors, and that was really exciting and hadn't previously been shown before. So again, just consistent. We still have a lot of replication to do here. I want to see the same effect in multiple ovarian cancer cell lines before we make any assumptions, but this is a really great jumping off point for future study. She also has just done this work in combination with Rob Schultz, who is really interested in bioinformatics and sequencing, looking at RNA sequencing analysis of the same OVCAR3 cells after 24 hours of atovaquone treatment. So basically saying, hey, we know what normal OVCAR3s look like from an RNA expression standpoint. Let's see what happens. What's changing in gene expression after atovaquone exposure? And again, very preliminary results, but we're starting to see some patterns in the types of pathways that are altered. So upregulation of genes involved in apoptosis, which is cell death, and again, upregulation of genes involved in the endoplasmic reticulum stress pathways and other molecular damage responses and immune processes. So kind of starting to tie this together, we now have other areas that we can potentially study to think about how these complicated interactions play into each other. So what she took home here and presented at the AACR meeting just last week was that it seems that several pathways of interest were in fact upregulated following atovaquone treatment, and that also atovaquone promotes the release of DAMPs, as she hypothesized. So what can we do next? Plenty. So we need to figure out a little bit more between the relationship between ER stress activation and immunogenic cell death, so we can best exploit that pathway as a novel explanation for why atovaquone could work as a sensitizer to immunotherapy. And then, of course, look at this in vivo. So mouse models and other models are accessible to us to potentially look at this more. There's other facets of the immune system that we're also studying. This will probably be what I get to talk about next year because we're just getting started here. But you remember when I showed you different cells in the immune system that I mentioned dendritic cells. And one of our other collaborators collaborators, Dr. Jenny Gumpers, who's awesome, and she's an immunologist here at the University of Wisconsin. She is looking at ways of supercharging dendritic cells by complexing them with a unique type of innate T lymphocyte called invariant natural killer T cells, so INKT cells. So basically, she's created her own immunotherapy, looking at dendritic cells and pairing them with the INKT cells. This is fascinating and something I did not know anything about until Dr. Gumpers and Dr. Patankar introduced me to it. And I think this is really exciting because there's a possibility we could actually create these complexes using patient ascites, so actually using immune cells harvested from a patient in particular and make immunotherapy from that patient's own, not tissue, but own fluids. So looking at the immune cells of that particular patient. So we're just getting started here. First of all, to see if T cell stimulating abilities can actually occur once we isolate cells from ovarian cancer patients' ascites, and then look at the different efficacy with atovaquone in mouse ovarian cancer models. So baby steps here, but I'm really excited about this next phase of research. Manish is always giving me a hard time because immunotherapy was definitely not my area of research interest. And it seems that every path this way has led me into I don't want to say the pit of immunotherapy because it's actually been a wonderful space to be in, but it's really forced me to learn. Immunology has changed a ton since I've been in medical school. So it's really forced me to expand my own knowledge in order to maximize the opportunities in our lab. Manish, I'm sure will have acknowledgements as well, and there'll be a whole lot of overlap. But I want to acknowledge our Cancer Center Director, Howard Bailey, who's always been incredibly supportive of our work. Everyone else here is a close collaborator. And then, of course, mentioning some funders, and WOCA has been one of them. So thank you very, very much for that. And I would be remiss if I didn't tell you, and I'll make sure this makes it to you all in an email too, so you don't have to try and copy this link. Actually, I'll also put the link in the chat. But the SGO and the Foundation for Women's Cancer together are putting on a scientific debrief from our annual meeting, which occurred in March. So the Society for GYN Oncology is where a lot of the new science and advances are presented. And what we want to do is bring this to patients, advocates, and caregivers who did not have an opportunity to attend the meeting. So we're going to do a debrief of some of the most exciting science discussed, and that's on Saturday, May 20th. And it's really meant for anybody. So I think we'll have providers on the call. I definitely think we'll have patients, family members, anyone who wants to learn more. Registration is open. It's completely free. And I will put this in the chat momentarily. I also have to show our amazing laboratory here. Most of the work I highlight today is Myra's work. She's in the front left here, and Rob, who is here in the black polo shirt. And this is our lab, including some already graduated students and family and friends, because of course we all know that without our support systems, we would be toast. I will end it there. And I bet Manish will talk, and then we can do probably questions at the end.
Yeah, thanks, Lisa.
I'll stop sharing. I think that worked.
Okay, now I'll share my slides. Switch. Okay, are you seeing my slides then? And not the presenter view, I think. Okay.
It looks great.
So perfect. So as Dr. Barriolhet, said, she focused more on the therapeutic projects that we have going in the lab, and I think I'll tell you a little bit about some of the diagnostic work, diagnostic test development work that we are also doing in our labs. And so this work is focused on CA-125, which you are all familiar with, and it's one of the, to me, it's like the only molecule that's out there to study, because I've been studying this since, I think, late 1990s, and I have continued to do work with this molecule. It's just a really, really fascinating molecule. And of course, the importance of this molecule is that it's CA125, and it has led to the CA125 test. But just to give you a brief summary of what this molecule is, if you haven't attended my previous lectures at VOCA on this. So CA125 that we all use as a test is really a tiny portion of this big molecule called MUC16. So MUC stands for mucin. And so this is a huge molecule that is expressed by ovarian cancer cells, but it's also expressed by normal cells, and especially in the gut and in our airways, it's expressed. But what happens is that ovarian tumors actually express it a lot more of this molecule, and therefore because it is expressed more. We are able to detect it, and an increase in the levels of this molecule is kind of thought of as a diagnostic for recurrence of the disease, right? So initially what we know is that the ovarian tumors express this molecule, and the molecule is actually displayed on the surface of the ovarian cancer cells. And then it somehow gets clipped, and we still don't know the exact mechanism by which this is getting clipped from the surface of the cells. So that scissor that you see, there is some kind of a molecular mechanism there that allows it to be kind of clipped. And once it is clipped, then it goes into the bloodstream, and that's where we detect it as the CA125 in the CA125 test. So that's how our test actually works right now, right? So again, this is another view of the molecule. It's a huge molecule. It's one of the biggest molecules in human biology. And there are, we have developed these antibodies, OC125 and M11. These are the two antibodies that are used in the serum CA125 test, and we use these antibodies to detect CA125. But really, what you're detecting is this big molecule that's in the blood. So we have a collaboration going on with Dr. Rebecca Whelan at the University of Kansas, and we are using some really high and proteomic work or protein analysis, let's say, methods to really get a sense of what this whole molecule is. And the CA125 test is really focused on those little portions of this MUC16 molecule. But can we now look at information that might be out there in the remaining parts of this molecule, and can we develop some tests based on that? So that's kind of a parallel work that we are doing right now. We are funded through the NIH on that work. But what I want to talk to you about is some other work that we are doing, which is really getting us to a point where we have now developed new technology to look at a different version of the CA125 assay. So right now, just to recap, the way we are working, the way the assay works is ovarian tumors express this MUC16 or CA125 on the surface. And then from there, it gets clipped, it goes into the bloodstream. And when we draw blood from patients, the CA125 is in the blood. And then there is an assay that we do where we use those two antibodies, that OC125 and M11 antibody, and that is used to capture the CA125 that is in the blood. One of those antibodies has a fluorescent or some kind of a marker. So the more of this marker bound, of this antibody with the marker bound to the cell, to the CA125 means there is more CA125. So that's how we kind of quantify how much CA125 is present in the blood. And so any elevation in the levels of the CA125 is what we are really concerned about, right? And in the test. So that's how the test typically test works as it is right now. But what happened was several years ago, and this was really going back to 2007, 2010, we were working on this molecule, and what we found out was that when we looked at the immune cells that were circulating in the blood, so these are white blood cells, WBCs, we saw that some of these immune cells that were coming from the patients actually were carrying this molecule, this MUC16 or CA125 on the surface, on their surface. So we were really amazed by that. We didn't know what was going on, and we did more work. And what we figured was on some of the white blood cells, not all of the white blood cells, but some of the white blood cells, there is a protein which is shown by this thing. I don't know if you can see my arrow, but you can see this protein. And that protein actually captures the CA125 that's out there in the blood, right? So the CA125 coming from the tumor goes into the blood, and that's where it meets these white blood cells that have this specific protein on it. And that protein just binds to CA125, captures it, and then the immune cell kind of carries it around in throughout the blood. So that then gave us this idea that maybe if we can look at the CA125 that's actually on the immune cells, that could be another potential test that we can develop for as a diagnostic for ovarian cancer. So we did a lot of work. We had a grant and from the NIH, and we did a lot of work, and came to after studying multiple patients, what we found out was, yeah, the MUC16 or the CA125 is present on the blood, on the white blood cells, but it's at a very, very low amount. It's only a few molecules here and there that are present on the immune cells, on the white blood cells. And so using our conventional approaches that we were using at that time, it was almost impossible to detect this on a consistent basis on the immune cells. And so the challenge was, how do we find this molecule that is present on this white blood cells? We know it is present, but we just don't have a way to find it, to detect it on the immune cells. So from like 2011, 2012, this whole project was kind of at a standstill, and we just couldn't do anything with it because there was just no way that we could detect the CA125 on the white blood cells with any kind of surety, right, with consistency. And so then what happened was just in, I think, 2014, late 2014 or early 2015, I just out of the blue, I get a call from two people who were in Boston. Their names are Petra Krolodat and Peter Hansen. And they had started a small biotech company. And they came to know about our work because we had published this. And they said, hey, we think, just out of the blue, they called me and they'd say, we know what's going on with you, with the challenges that you're facing. And we think that the challenge is that you just can't find it. You just don't have a way to find MUC16 on the white blood cells. And I said, yeah, absolutely. That's our biggest challenge. So they said, we have a solution for that. And so what they were doing was they had developed this technology that uses gold nanoparticles. So these are really, really tiny gold particles. And these particles kind of exhibit a physical phenomena called surface plasmon resonance, or SPR. So because of this SPR, they can really shine, and if you put light on them, they can really shine and be intensely bright. And I'll show you in just a few minutes. I'll show you some pictures of what that intensely bright spec looks like. But they said that, look, we have developed these gold nanoparticles, and we have developed some technologies, which is pretty rudimentary at this point. But if you are willing to collaborate, we can take this project further and develop a whole technology based on this. And I said, of course, that's exactly what we are looking for. So again, what we want to do with this technology is this: if this is the surface of the white blood cell, and that's that protein that is holding on to MUC16, this is the protein called Siglec-9 that is holding on to or capturing MUC16 on its surface. We are going to use these gold nanoparticles to find out how much of this MUC16 is present on the immune cell, right? So that's kind of the assay that we want. We couldn't do this using our existing technology, but now with the help from P&P Corporation, that's what we are focusing on. And I think I'll show you some of the advances that we have made. made on this thing. So this is really a collaboration with Peter Hansen, Petra Krolodat, and Herman Gonzalez from PNP Corporation. They have the technology, and it's a kind of a wider collaboration that we have with Dan Kramer and Kevin Elias at Harvard and Brigham Women's Hospital, and also Jan Zhu Chen, who is a immunologist at MIT. So it's kind of a part of a bigger collaboration that we have going with this whole group. So this is what we have now. So we one time, I think in 2018 or 2019, we had a VOCA of actual in-person lab visit for VOCA. And so people who came to that visit may remember this microscope kind of a device on the left. So this is what we had in the lab at that point, and we were using this. This is the device that was developed by PNP Corporation. And this device now is geared to really look at those gold nanoparticles, that intense light that comes from these gold nanoparticles. And I'll show you pictures of that in just a bit. And so we have done a lot of this initial work using this microscope. And that's called the breadboard prototype. That's really the initial kind of a rough microscope that they developed, and we had it in our lab. Then they took it back because they said, hey, the data looks promising. We want to develop an even better unit. And this is kind of the prototype that they are working on right now. It's all in a box, as you can see. And basically the sample goes up at the top where this door is open. And the whole idea now is the sample goes there. This will be white blood cell samples that we have labeled with those gold nanoparticles. And we shut the door, and pretty much it's an automated system. It will go in, look at the white blood cells, look at how much of these gold nanoparticles are bound to are present on the white blood cells. Because the more gold nanoparticles are present, it means there is more CA125 that is on the white blood cells. And so the more gold nanoparticles we see, that means that there is more CA125, and then that kind of becomes like the diagnostic. So that's what we have. We don't have the microscope right now, but we are trying to get this newer version eventually that we will then start working with. So this is what I was talking about. These are the gold nanoparticles. You can see how intensely bright these molecules are. So in the background, you will see that hazy kind of background is the cell. So there are 3 cells we are showing in the top image, and there are some other cells in the background as well, but we are focusing on these 3 cells. And those little tiny specks that you see, those are the gold nanoparticles. And when you have these gold nanoparticles, that is basically kind of like a surrogate kind of measurement of how much CA125 is present on these immune cells. So you can see these little tiny specks, and the machine then can kind of goes in and counts how many of these dots we have per cell. The more dots you have, the more CA125 you have on these cells. So it's these 3 cells are then magnified in the bottom 3 pictures. So again, you see that hazy background, that's the cell. And then the fluorescent, the really intensely bright dots, those are the gold nanoparticles, right? So that's we have been getting these images now, and they look really, really beautiful. And our computer that is hooked to this machine is going in, and we have trained the computer to look for these dots and spit out a number of how many of these dots are present per cell in any given white blood cell sample. And that's kind of the way we are approaching this development of this new diagnostic test. So again, this is what we are trying to do. The tumor produces MUC16 or CA125. It goes in, binds to the immune cells, and we are trying to detect how much of this immune CA125 is present on the white blood cells of the immune cells. Now, what we have shown in the past as well is that this protein that captures the CA125, it's present on not all of the immune cells, but quite a few of the subsets of immune cells that you typically see in the blood. So for example, T cells, NK cells, NKT cells, B cells, monocytes. These are the different subsets of white blood cells that we typically see in the blood. And so some of these you will see that some of the, like the T cells and the NK cells, have kind of lower levels of these dots on them, whereas the B cells and the monocytes have higher levels. So it is harder to find these dots on the NK cells and especially on the T cells. But because this technology, this gold nanoparticles are so intensely bright, even if we don't have a whole lot on the T cells, we can still find them. We can still find these dots by using this new microscope. So that's, we've published this, and we have shown in our initial group of about 20 ovarian cancer patients and 20 controls, we have shown that the amount of CA125 that is present on the immune cells is higher than healthy controls. So we have some preliminary data showing that it is on the higher side, and we are now really expanding our studies with more patients, trying to see how, if we can get more of these data, and then those data then can be used for development of a diagnostic test. So what PNP Corporation has done is they have now got a collaboration going with a hospital in Madrid. So this is Hospital Clínico San Carlos, and Dr. Antonio Casado, who is a gynonc surgeon there. He is our collaborator. And what they are doing in Madrid is they are collecting blood samples from patients, from ovarian cancer patients, as they are going through treatment. And so we have got enrolled 33 patients, and we are continuing to collect blood samples from them. So Dr. Casado is doing that at Madrid. And then once the blood samples are collected, they take the white blood cells and then freeze them down. And the whole idea now is that these frozen white blood cell samples, once we get this new instrument back in our laboratory, that those frozen samples will be shipped from Madrid to us, and then we will do our analysis in our lab here at UW using this new instrument. But we have these really good bank of samples that are, we call them longitudinal samples. So every time the patients come back for treatment or other things, we collect blood samples from them, and those samples are now stored in Madrid, and then we will eventually get those samples here and do the analysis here. So the folks at PNP have taken a few of these samples. They are from about 5 to 9 patients, and they have done their own independent analysis on this. So this is just a small group of patient samples. So they have done some analysis on this, which looks promising. We are quite far away, of course, from developing a test yet. We need to do a lot of this work on a lot of samples right now, but the data that we are seeing is pretty promising. So, and this, I'll show you data from 2 of those 5 patients just to give you an idea what's going on. So in this plot, the blue line is this blue solid line, are CA125 values using our conventional serum CA125 test, right? So this patient underwent chemotherapy, and you'll see that there is a big drop in the serum CA125 levels. So that blue line just drops completely down, right? As this patient was going through, we were collecting samples from this patient, and those white blood cell samples that we were collecting were then used to look at how much CA125 was present on the white blood cells of these patients. And the red line is now showing the amount of CA125 on the white blood cells from this patient using that old microscope that PNP has developed. So this is actually counting how many of those gold nanoparticle, those dots are present on the immune cells, and this is specifically on the T cells, right? So this is that T cell population that typically has very, very low levels of the dots. So even that population, what we are seeing is there is actually an increase in the CA125 that is present on those T cells. And that increase, if you compare that increase with the blue line, which is your CA125 test, that increase is way before. I mean, the blue line is pretty much uh, at the bottomed out, whereas we now see an increase in the CA125 that is present on the white blood cells. Even when the CA125, serum CA125 has bottomed out. This is interesting because if this is true and if this is proven, then that means that our new test can start detecting CA125 on the immune cells way before the serum CA125 test can show a positive result. There is another patient, again, kind of a similar profile. CA125 drops down and this patient was going through a treatment regimen, so that the CA125 drops down. But then if you look at the red line, this is looking again at the PNPs or the CA125 that's present on the T cells. That has started going up even when the CA125 serum levels have dropped our baseline. So this is really initial data. It's very interesting data. And again, I want to caution everybody that we don't have a test. There's a lot more work to be done on this, but the data looks interesting. And it's because of this interesting data that we are now in negotiations with WARF, which is a patenting body here on campus. And they are interested in providing us some seed money, some seed funding to develop the technology a little bit more, but also then to get those samples from Madrid. And we'll also be getting some samples from Harvard and Brigham Women's Hospital, and our own samples that we collect from patients here at UW. So that way we'll have a bank of samples that we can now test using that new the instrument that we get from PNP, and really do these studies that will be kind of the foundational work for the and if it works out, and if the data is consistent, then these foundational data will be used for the development of the new test. So we are in negotiations with WARF to secure this funding. PNP has also been in negotiations with another industry partner that has a tremendous knowledge on developing this device, that box, because that needs some more engineering and some more development. So they are in negotiations with another company as well. So we are in a really good position right now. Once we secure some funding, we have the samples, which is the most precious thing. Funding, is not as precious really as the samples, because over in the world, there's hardly any people that are actually storing the white blood cells from patients. And through our links with Brigham Women's and the Madrid hospital and our own samples, we have a really good bank of these samples. So we'll test them here once we get some funding from WARF. So that's kind of the update on that project. And again, as Lisa said, this is this is all happening because we have some really excellent people in the lab. I should mention Nisha Boon-Patrawong, who is our postdoc, and she's been working on a bunch of projects, including this MUC16 project, but also some of the work that we are doing on atovaquone. And Rob Schulz is our lab manager. Sejal is a grad student, a PhD student in the lab. Lisa already mentioned Myra, who is our MD PhD student, and Pratichi is my master's student, who is working on MUC16 biochemistry project. Then we have our fellow, Dr. Matthew Vega. We didn't get a chance to talk about his work here, but he is working on another cancer biomarker project. And then our undergrads, Daniel, Samantha, Tyson, and Abraham. So it's a team effort that is allowing us to do all of this work. And then one exciting news I want to share with you is our department, OBGYN, has decided to hire Dr. Maria Virambrales Munoz. She will be our newest faculty member in the department, and she is a bioengineer by training. And what she will be doing when she starts her lab here in our department is she will be developing models of the fallopian tube and ovarian cancer. These will be in vitro models, so using bioengineering concepts. And so once she develops these models, we'll be able to do a whole lot of different studies. We'll be able to test different drugs and also understand the biology of ovarian cancer. So that's really exciting for us. And we're looking forward to having Maria join our department and working with her in the department. She will be, her lab will be, kind of space that Lisa and I share. So we'll all be here in this together. So I'll stop there, and hopefully I've left some time for questions.
All right, great. Thank you both so much.
Yeah.
All right, we will open it up to questions now. If you have any questions, feel free to unmute yourself and ask. Otherwise, you can put them in the chat as well, and I can moderate them, whatever you prefer.
So there's a question from, a couple of questions. Oh, actually from Kathy.
Oh. Let me see here.
Would you be able to see the highlights of the cells in MUC16 for an ovarian cancer patient with low levels of CA-125? Yeah, that is exactly the point, Kathy. It's a really great question. So typically with the conventional CA-125 assay, if your level of CA-125 is very low, then that assay is not really geared for detecting such low levels. But our, if with the gold nanoparticles, because the detection can be at such a low level, we think that we'll be able to see MUC16 on immune cells even when the serum CA-125 levels are low. So it's all like the sensitivity of this assay is really tremendous. That's a good question.
Any other questions? I know we got a couple minutes left. Don't see any in the chat.
I did post that information about the scientific debrief for anyone who would like to join us in May.
Great, and we'll send that out. We'll send a recap email out next week, and we can include that too, Dr. Barriolhet. And
Perfect.
People, we can promote it on social media as well.
Awesome, thank you.
Of course. Any other questions? Well, thank you all so much. This was a great kickoff to our week of webinars for our Survivors Brunch. Thank you, Dr. Patankar and Dr. Barriolhet. I know you both are extremely busy, so we really greatly appreciate you taking the time to join us today. And then I hope everyone has a wonderful day. Thank you.
Have a great day. Thank you.